Now, before we move on to the real content of validation, isn't it important to also understand what are the prerequisites, what you must do before you start thinking about the validation activity? It is very, very essential. If you make a proper planning of the validation, you know, you will have lesser difficulties in completing the validation. So it is very important, the first and foremost, about having the suitable test procedure. One cannot perform the validation without having the test procedure. But when I say test procedure, the meaning of having a suitable test procedure is very, very important. So how you can say my test procedure is really suitable in its own way? Whether it is, you may say, okay, now my test procedure has all required conditions of the chromatography conditions. For example, my test procedure talks about the calculation part. My test procedure talks about the representative chromatograms. My test procedure talks about preparation of standard sample in very, very detailed way. My test procedure talks about how to conduct the analysis, what should be the sequence of analysis, etc. So that is called as the suitability of the test procedure. You can, the one can say, okay, I have everything, but if the test procedure is not signed by the authorities, maybe your designees like the quality assurance department or your department heads or the reviewer, it will not become the official test procedure for me. So having all these details, plus it is getting signed off from the your authorities according to your SOP, I think that makes the test procedure suitable and you can use that test procedure for the validation. Now, here is the chicken and egg concept now. You may be having point in your mind, okay, Bhaskar, I understand that the test procedure should be suitably signed off, should be suitable one, but how one can confirm without validating it whether the test procedure is really suitable or not? You are saying your test procedure must be suitable for the validation. But look, I have not conducted its validation. And how I can say that my test procedure is a suitable one for conducting the validation? It's a chicken and egg phenomenon. Which one is first? So to get the confidence onto your test procedure, you may not have to complete the entire validation, but there is something you can have a mini validation concept. Mini validation means what? You are not going to perform the complete validation as part of your method development exercise, but at least you are going to touch upon few of the very critical aspects of your validation method performance parameters, like resolution impact, like the specificity of your test procedure, like the accuracy of your test procedures. So these are some parameters you have to perform before you start validating it. You cannot just say, okay, I got the chromatography condition and let us begin the validation. Performing the mini validation is probably the first step in making the test procedure. How many of you have this practice in the place? Type me in the chat box if you follow this practice of performing the mini validation. Maybe 2, 3 different parameters, not in complete way, but you have that practice. It is very, very important. Yeah, many of you have this practice, and it is very important to have this practice. If someone does not have this practice, for some reason, please talk to your senior management, talk to your seniors, and get this as your activity. Before you conduct the mini validation, you never supposed to go for the validation activity. So the test procedure can be defined once you have the mini validation done, isn't it? The second important document you must look for is the specification. Yeah, the workability study. Thank you so much, Jaiti. So what is meant by the specification? The specification is the list of your test, the list of your test that is going to determine the product's quality. It may be about physical test, chemical test, it may be about the performance characteristic test like dissolution or disintegration time. So your specification must be ready and available before you start validation. And it is not only about the one specification, but in the drug products, there could be two different specification available. The first one is release specification, and the second one is the shelf life specification. So how one can prepare the release specification? See, the release specification is the specification which is going to control onto your process, which is going to control your process parameters. And what is the shelf life specification? The shelf life specification is going to meet the regulatory requirements. The shelf life specification is going to meet your monographs requirement in case if your drug product is official. So to give an example, let us say you have a impurity A as your part of the specification. Now this impurity A is a degradant. Or to observe that this impurity A is a degradant. And to make sure that the impurity A is remaining within the specification, you are going to make sure that our process will be designed in such a way that impurity A content will be as minimum as possible. Right? Let us say that your monograph requirement, your regulatory requirement for this impurity A is not more than 1%. Right? The monograph says the impurity A should not be more than 1%. So what is going to be your shelf life specification for this impurity? Not more than 1%, right? This is going to be your shelf life specification. But when you talk about now the release specification, are you going to have the same limit for the release specification? Type yes or no in the chat box. Are you going to keep not more than 1% as the impurity A limit for the release time? Absolutely not. Because you know that this is the impurity which is degrading in the nature. It's a degradation impurity, and I have a 24 months to go. So I will control the process, the manufacturing process in such a way that the impurity content will be as low as possible so that I will get the buffer space to make sure that the impurity will not fall out of the spec at the end of 24 months. And then you will validate your process, isn't it? You will say, okay, now if I have the control temperature, if I have the this and that control, what is the best impurity A I can get at the release time? And you will say, fantastic, I can able to have the impurity A content into my batch not more than 0.2%, not more than 0.2%. So based on to your stability trend, the rate of degradation, you say, okay, now if I have the 0.5% at the beginning, probably it will not reach beyond 1% based on to the rate of degradation. So let us keep not more than 0.5% as our release peak, right? Not more than 0.5 as our release peak and not more than 1% as our shelf life or the stability peak. So all this specification, needs to be available in hand. How many of you got an idea how the release and shelf life specification can be designed for your product? Type me in the chat box if you now got some idea about designing of the release and shelf life specification. It is very important to understand the logic behind the setting the different specification. So understand that your specification is signed and available for you. Now, once you have the test procedure, once you have the specification in the hand, without sample, is it possible for you to think about conducting the validation? Yes or no? So you should have a sample in the hand. And when I talk about the test sample, when I talk about the sample, when I talk about the test sample, what characteristics that you are going to look into the test sample? What characteristic that you are going to look into the test sample? The first one is whether it's meeting the specification or not. The second one is, okay, now whether the test sample is homogeneous in the nature or not. In case if there is a inbuilt variability inside your test sample, that test sample is not going to be suitable to be used for the validation. So the test sample must be homogeneous in the nature. The third important characteristic could be the quantity available. See, you need, let us say, around 100 gram of the API or 1000 number of tablets for the validation. So in case if there is no enough quantity available, there is no meaning of taking that particular batch for the validation. And also, it is very important to understand that, what is the expiry or the shelf life or the retest date for the sample that you are going to use. Have the enough amount of shelf life available so that, because you may have to run the validation for 1 month, 2 months, or sometime maybe more than that because of the complexity and number of test procedure, number of test like assay, dissolution, RS, and whatnot. Okay, there could be the longer period required for the validation. And also think about the method transfer too. It's always advisable, not possible every time, but it's always advisable to use the sample which is used for the validation for the method transfer also, because you have some confidence onto the batch that you have used for the validation. So in case if you have enough amount of quantity available, it's a good idea to use the same sample for the method transfer. Because look here now, if you are conducting the validation at your R&D center, your R&D is not going to use the method for the lifetime of the product. So where the method is going to use, it will be used at quality control lab. And you need to transfer the method to the quality control unit. So if you can have the same sample which is used for the validation, it's a good idea. How many of you think it's a good idea? Type me in the chat box the common sample for validation and the method transfer to avoid lot many surprises. Because otherwise, if you change the batch and for some reason, if the batch is not homogeneous, and you will unnecessarily stuck in the method transfer activity. It's very important. Okay, then how one can neglect the standard availability? Now, when I talk about the standards availability, it is not only about the just having the one file of the standard inside your standard storage area, but it is all about of the suitable purity or potency. It is all about the suitable quantity. It is all about whether there is a suitable time period available for its usage or not. Just imagine if you start performing the validation and your standard gets expired tomorrow. I hope this will not happen with you. But then it's completely havoc, right? You cannot perform the validation tomorrow. So it's also very important to have the standard with the adequate usage period. Also think about whether you have the right standard source. What is meant by the right standard source? Now, sometimes you may have the multiple grade of the standards, USP, EP, in-house, and whatnot. So understand that for which geography you are validating the product. It is always advisable. It is not something which is very, very straightforward and that must be the requirement. But it is always advisable to use the standard of USP for the US application. It is always advisable to use the EP standard for the European countries applications. And in case if you do not have the USP standard, then you can substitute the EP standard for the method validation and vice versa. Even though you can use the in-house standards too. If you do not have the standards provided by either USP or EP, you can have your own in-house, or you can have the standards manufactured from the third party like LCGC, like Merck. So all the standards are also well acceptable in case if they have the suitable backup data, suitable characterization data like NMR, mass, their potency calculation, right? Maybe their CHNO analysis, maybe that is going to talk about the residual solvent, the water content analysis, so that you are very much sure about the potency of the standard that you are going to use. Now, is it a network problem? How many of you can see me and hear me clearly? Type yes me in the chat box. Yeah. Okay. So I think majority of the people can see me and hear me. Yeah. Thank you so much. It's great to know that. Thank you. Thank you for your response. So understand the standards, and also when it comes to standard availability, it is also important to understand, how many standard vials are available with me. How many standards vial available with me? Sometimes there are practices available that you can use the standard vial for only once. You can use the standard vial for only once because the standard is highly hygroscopic in the nature. So if the standard is highly hygroscopic in the nature, probably your SOP will say use one vial at a time. And then you will calculate, okay, each vial has 500 milligrams. So how much standard is required for me? I will require, let us say, 2 grams. And I have 10 vials available. 10 into 500 becomes 5 grams. It looks to be sufficient. but you can only use those 10 vials for 10 times. But if you have to use the standard for 20 times, then because you're just using 100 milligram, but the remaining 400 milligram have to be discarded. So for that reason, it is very, very important. It is very, very important to understand the usability of the standard available. Yeah, how much standard actually you can use. If there are multiple vials, that doesn't mean that that is the entire standard you can use. How many of you have faced this challenge? Type me in the chat box. You calculated the standard availability based on to the exact quantity, but you missed on understanding the vial usage time, the period. It is very, very crucial for the highly hygroscopic working standards or the reference standard. In case if your standard is supposed to be not used for only once for a month's time, then probably you can have the better time. I hope you understand the complete requirement of test procedure, specification, test, and the standard. Type yes in the chat box. Before we move on to the placebo for drug product, right? It is also very important to have the suitable quantity of the placebo. Now the question over here, okay, how one has to make the placebo? How one has to make the placebo? Do you need to have the processed placebo, or can you have just the handmade mixed placebo? The processed placebo should be preferred. Why? Because the processed placebo is actually manufactured by following the all procedures and the steps as like your drug product. So in case if there are certain characteristics of the placebo may get impacted because of your blending time, maybe because of the shifting time, anything which may not be, actually reflected those characteristics if you make the placebo by just hand mixing inside the lab, the polybag mixing, right? So most of the times, in case if your placebo content is very, very low, in case if your placebo content is very, very low, the placebo is nothing but if you have a paracetamol tablet, if you have a paracetamol tablet with the label claim of 500 milligram, you have 500 milligram, but your tablet weigh around 1 gram. Your tablet is, the tablet's weight is 1 gram, the 1000 milligram. So whatever another 500 milligram present into your tablet is called as the placebo. So all the excipients used to compress the tablet into a suitable form is called as the placebo. So this placebo is very, very essential, especially for the drug products like tablets, creams, ointment, or injectables. Without having the placebo, you may not be able to perform the validation. So it is very important to have the process placebo. Now let me explain you. There could be chance that you may have the placebo which is just handmade. The challenge is if your placebo content is very, very high into the drug sub product. Let us take an example of the very low label claim dosage forms like oral contraceptives. Now, this drug products has very, very low amount of content. Active content is very in terms of microgram. So the placebo content is very huge. Now, in those situations, as your placebo matrix is very huge, the slight impact, the slight change in placebo composition can also impact onto your placebo characteristics. Your placebo chromatogram may look different. It is very important for that reason. You have to make the process placebo. How many of you have faced the change in the placebo's pattern during the validation? You saw some different pattern during development, right? For the placebo, there were only 2 peaks coming up out of the placebo. And when you started the validation, you start seeing 10 different peaks in the placebo. How many of you have gone through this? I have gone through this. So this may be the possibility because you are not retaining the entire characteristics of the placebo. So it is very important to have the placebo manufactured by the process way, so that it is not very unnecessarily for you to investigate the placebo differences. The chemical and reagent is absolutely one part which you are not going to miss on. The another important point is instruments and equipments. So most of the times your R&D may be in good contact with the quality control team, and I hope that all instruments available at R&D will also be available at the quality control. but it's very important to give a thought on it, whether do I have all the required instruments at my quality control lab too? Do I have all the required equipments? Sometimes, if I'm not wrong, we were working onto the antacid tablet, and as a part of antacid tablet, there were some inorganic APIs. I think magnesium hydroxide or something, right? Is the right magnesium hydroxide, and there is one more as a part of your antacid tablet. So this magnesium hydroxide, uh, and there is simethicone present into it, right? There is simethicone present into it. And to quantify the simethicone, I think it is the liquid extraction technique, aluminum oxide and magnesium hydroxide. Thank you so much, Nilesh. So we used to conduct the estimation of simethicone or dimethicone. I do not remember exactly, but I think simethicone was the active part of it by using FDIR. How many of you have done the simethicone by IR? Type me in the chat box. Ketan, you did it. Isn't it? And how many of you know that it is very hard to recover the simethicone out of your antacid tablet? How many of you face the recovery challenges? Type me in the chat box. Or if someone is still facing, type me in the chat box if you're facing that recovery challenge. So what we did as a part of our development, it requires a very vigorous shaking. You need to take the organic layer, which is immiscible with the water. I think we have taken the chloroform, or toluene as a layer, as organic solvent, and then hydrochloric acid as the aqueous layer. So we used to shake for a lot of time, because the issue is, the male scientist used to get better assay, and the female scientist used to get the lower assay. And then again, there is hand-to-hand variation. And we completely understand that this vigorous shaking is very, very important to get the recovery correct. This physical shaking is very, very important to get this recovery correct. And upon understanding the challenge, we introduce something called as the rotary shaker. Now, this is a shaker which shakes the flask in the not in the this direction, but in the vertical direction. And because of that, the shaking becomes very, very vigorous. Now we got the equipment in place. Yeah, we got the equipment in place at the R&D, but we have missed to inform to the quality control team that, okay, now for antacid tablet, you need this very special equipment. It was very small equipment, by the way, but we missed on to that part. So sometimes, out of this urgency, we sometimes miss out this. It is very important to have the constant contact with your quality control team onto your product development or the method development. And then we have the 11th hour rush. So we got our R&D equipment installed into the quality control unit, and then the method transfer and followed by exhibit batches was taken. But it was little chaos and unnecessary to become panic. So to just avoid that, it is very important to have one-to-one meeting with your quality control counterpart. Look here, these are the equipments required. This is the test procedure, and let them have a chance to go through your entire complete test procedure. How many of you have this procedure in the place that you meet with the quality control team, right? If you're part of the method transfer especially, and you understand the method, you understand the requirement very well, type me in the chat box if you have that practice in the place, right? And if you do not have, please make this as a practice. It is going to help you out to avoid this 11th hour rush, the like we had during the antacid method transfer. That tech transfer meetings, Bala, absolutely. So, and I'm sure that you must be taking care of the qualification and the calibration of the instrument. The instrument operation range is very, very important. Now, when I talk about the instrument operation ranges, let me give you an example. Let us say you have a gas chromatography test procedure for residual solvent. And it happened with our team some time ago. Okay. And most of the times for the gas chromatography, we used to start from the 40 degrees Celsius as a column oven temperature, the 40, the 40. But for some reason, for the current product that we are handling and working with, which we could not get the required resolution. And because of that, we started the column temperature with 35 degrees Celsius. With 35 degrees Celsius. Now we missed on to passing this information to the QC. With saying, okay, now there is a GC available of the same adjuvant 6890 with a similar kind of headspace, nothing to worry. Let us begin the transfer. But when we prepared the protocol and then we realized that, look at here now, your temperature is 35 degrees Celsius. And if you look the calibration part of the gas chromatography available at quality control, their temperature calibration range actually starts from 40 degrees Celsius. So how they are going to use the GC instrument for 35 degrees Celsius? What is your say? Can they use the GC for 35? Type yes or no in the chat box. Okay. And then again, that's the the running here and there, making the protocol, taking the incident, the deviation, completing the calibration part for at least temperature and etc. But that can be avoided, right? That can. How many of you understand the instrument operating range? Why it is so important? Why it is so important to also look into your methods parameter and the instruments operating range. Type GI if you got it. Type GI if you got it, because it's very important, and I hope that you will not miss onto it, but it can get missed out. Thank you so much. The concentration matrix is a very important point, and this is the point where you are going to make the concentration of your standard solution, your test solution, your impurity stocks, etc. It is very important that this particular concept that you must implement at your work area. See, it's my own practice. When I was working on the validation, I used to make the concentration matrix before I think about the protocol preparation and stuff like that. I make the concentration matrix even during the execution of the development experiments. I used to make the concentration matrix even during setting and designing the STP, the standard test procedure. It is very, very important because you need to understand there is a correlation between your standard concentration and the specification of the impurities. And for that reason, the concentration matrix really helps a lot. Even if you think about performing the validation with the multiple parameters like precision, accuracy, linearity. Now, if it is assay, it can be quite easy and you can manage the concentration changes. But just think about in case if it is a related substances. In case if there are two different specifications to be followed up and there are 10 different impurities, is it really easy to calculate the concentration on the same day during preparation of the protocol? It is not. And hence, I strongly recommend that you follow the concept which is called as the preparation of the concentration matrix. So how many of you know this concentration matrix, or how many of you are following this concentration matrix concept? Yeah. Type me in the chat box if you are using the concentration matrix. For some reason, if someone is not using that, we will talk about the concentration matrix preparation. Yeah. During the presentation, it's very, very important. So concentration matrix means, let us say you have a linearity parameters to be executed. What is that? Linearity. So what are the 5 different levels that you are going to choose as a part of linearity? You say 50%, 75, 100, 125, and 150%. But at 75, the 50 and 100% levels, you have to identify their actual concentration in terms of, let us say, microgram per ml, in terms of ppm. And then you realize, okay, now my standard concentration is 200 ppm. My standard concentration is 200 ppm. So what is the 50% of 200 ppm? Type in the chat box. So that becomes your 50% concentration. Now what is the 75% of the 200 ppm? Now that becomes your 75% level. And so on, you calculate the concentration of all these 5 linearity levels. So this is called as the concentration matrix. This is called as the concentration matrix. I hope all of you now understand what is meant by concentration matrix. Type you in the chat box if you understand what is really mean of the concentration matrix. And how one can deny the protocol necessity. I think that is one of the very important stage, the milestone you think about performing the validation. The test, procedure, the sample, the specification, matrix, etc. is just the secondary. But very important is your validation protocol. Unless and until you prepare the validation protocol, you really cannot understand what I have to do as a part of this validation activity. Do I need to perform LOD, LOQ? Do I need to perform accuracy? If yes, what are the preparation? What are the levels? All have to be in detailed into the protocol. So it is very important for everyone performing the validation, to understand the protocol preparation, to understand the protocol preparation. So it is very, very essential part. It can be about understanding the scope of the protocol preparation. It is about understanding the purpose of preparing the protocol. It is about understanding the which team is going to part of the validation activity. What are the instrument and equipment will be used for this particular validation activity? What sample, what standard will be used during this validation activity? What is the test procedure that you are going to use for this validation activity? What is the specification that you are going to use? And then finally, the respective standard and sample preparation for all the test parameters, like how the precision is going to get executed, how the standard will be prepared, a very detailed way, right? The way so and so milligram of the paracetamol diluted to so and so ml, filter it, centrifuge it, use so and so filter paper. So all the detail has to be given in the protocol itself. And then you can have the preparation of the sample, etc. So it is very important that your protocol must be well written. Just imagine that, okay, you are not going to perform the validation by yourself if you are making the protocol, but someone who do not have much experience, someone who do not done the validation earlier, if the protocol is given to that person, whether he or she able to perform the activity or not. And until and unless you get the answer, yes, 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 start looking at, making your protocol better and better and better. So that is the way you must look at the protocol. Protocol must not be prepared by thinking that, okay, now someone is well experienced, and if I miss on giving some details, it's okay, she or she will take care of that. You are making the gross mistake. The protocol must be prepared by keeping in the mind that the person who is completely fresh for the activity, who has never done the validation, now she or she is going to perform this validation. And what details she or she may look for. And then you prepare the protocol, and I promise you, you will get the best protocol in the place. The calculation sheet is very important, right? You cannot calculate the end result just manually, isn't it? Because there are a number of calculations you have to undertake and perform, and it becomes a very, very Herculean task. So you may perform the experiment quickly, okay? And you may have to invest a lot of time doing the calculations. And for that reason, it is a very good idea to have the automated calculation sheets. And I will talk about the preparation of this automated calculation sheets also. How many of you are using the automated calculation sheets? And how many of you are not using the automated calculation sheet? Type yes if you're using it, type no if you're not using it. I can say most of you are using it, some of you are not using it. Please understand the necessity of the calculation sheets. It is very, very easy to prepare, and it is once it's validated, understand this, you cannot just prepare the calculation sheet and start using it. You also need to validate your spreadsheet, your Excel sheet. And there are ways to do that. There are ways to do that. But once you conduct the validation, then look here now, you do not have to worry about anything. No one is going to change your calculation formula. No one is going to, make your calculation sheet which is going to give some erratic wrong result. Once you lock your calculation sheet, right, with the password protected, and that should be a part of your SOP, that who have the protocol, who have the password for the Excel sheet. And most of the times, the IT person or the QA person will be the right one. It is not the end user. If you are making the spreadsheet, if you are validating the spreadsheet, it's not you supposed to control that. The quality assurance is the right person, right team to control the password, right? The quality assurance, because it's look at here now. Sometimes out of some concerns, the team may change some of the calculations given inside the calculation sheet. So to avoid this such kind of incident, mishappenings, you can have the protocol along with the QA team. So in case if you have the password with the QA team, it's perfect. But in case if you do not have the password with the QA, you should think about changing your SOP. How many of you keep the password with the QA for all your Excel sheet and the spreadsheets? Type QA in the chat box if you have the password with the QA. If you keep the password with IT, type IT in the chat box. If you keep the password with quality control, type QC in the chat box. Yeah. So, okay. So some of you have with the QC, please think of it second time. And then finally, you get everything in the right place now. But if there is no execution team available, how one can execute the validation? So I give the prime importance to the execution team. I give the prime importance to the execution team, that the execution team should be laser focused, and the execution team must well trained, well understanding about the complete validation process. Not only that, the validation team must also aware about the consequences of something doing wrong during the validation. See, there could be situations where your recovery gets failed. There could be some experiment that has completely gone bad. You should not worry about that, but you should worry about, investigating those failures with the right systems. You may have to raise incident or the lab deviations for some reason, but document it, raise the deviation, raise the incident, have the appropriate kappa in the place, and then you go forward. Otherwise, if you just hide something out of your timelines, out of your boss, out of saying, okay, I'm very much scared now, what people will think about me, this is a silly mistake I have done, don't worry about anything. Don't think about today's moment. Think about what are the consequences this mistake will have in the future. And you will find there are numerous examples where, because of this kind of situation, the validation got completely rejected by the regulatory bodies. So it is very important for you to understand that it is not only about the matter of completing the validation, but it is about completing the validation activity with all required data integrity. I hope you understand. This type yes in the chat box if you understand why I'm saying the execution team is so important. Excellent. Thank you so much. And we just covered off the the first part of the presentation.